Hello, everybody. My name is Kit Ung from the Giggly Immunology Lab, and today I will present to you the importance of Qa-1b for the generation of adaptive immunity during Toxoplasma gondii infection. So let's get started. What is Toxoplasma gondii? Toxoplasma gondii is an eukaryotic apicomplexan protozoan, obligate intracellular parasite of animals. On the right, we can see the morphology of Toxoplasma gondii. It is 2 microns in width and 6 microns in length. This parasite infects nearly 30% of people worldwide. And this picture was taken from Dr. Boothroyd's lab in 2000. Here we can see the parasite's life cycle. T. gondii's definitive host is the cat, where unsporulated oocysts are passed into feces, where it can contaminate water or vegetables or uncooked meat. And when that enters the intermediate host, it releases sporozoites and differentiates into tachyozoites, where tachyozoites are going to attack regular cells. And when the immune system gets activated, the tachyozoites get stressed and they undergo a life stage switch into bradiozoites, and they form tissue cysts. These tissue cysts will remain inside the body until they die or and eaten by other organisms that pass the parasite around until they get back to its definitive host, the cat, where their molecule in the cat called linoleic acid, which triggers the parasite to go back into its sexual stage. This disease that Toxoplasma gondii causes is called Toxoplasmosis. This is a seropositive infection, meaning that people infected with this disease will have it for life. And we can see here's a map dating from 2009 showing the seroprevalence of this disease. In immunocompetent people, Toxoplasma gondii is normally asymptomatic, and there may be some behavioral changes as shown very recently in public years. But in the negative effects of Toxoplasma gondii are observed in pregnant women and immunocompromised people, where in pregnant women, the fetus may get infected by this parasite and may cause hydrocephaly, damage eye congenital toxoplasmosis start, or even worse, spontaneous abortion. Whereas in immunocompromised people, it may lead to toxoplasma encephalitis and even death. As of 2020, there's no cure available for Toxoplasma gondii infection, and unfortunately, there's no therapy or vaccine that is 100% effective at preventing or clearing T. gondii infection. So to understand this parasite, we need to know what the immune response is to this parasite. Well, the immune response of T. gondii is a very diverse one. It's controlled by CD8 T cells and the production of the cytokine interferon gamma. And there are very major gaps of knowledge of what regulates the development of the CD8 plus T cell response to control T. gondii infection. This is the mechanism of the immune response to Toxoplasma gondii. Antigen-presenting cells, APC, mainly dendritic cells, produce IL-12, a cytokine, via toll-like receptors, and this is shown in Heufler et al. In 1996. CD8 plus T cells then produce the cytokine interferon gamma due to IL-12 production. Now, after that, cytokines, as we know, they target infected cells and lead to apoptosis via many mechanisms. And what interferon gamma does in particular is that it starves the parasite and kills it in the inside of the cell it's living in. And this was shown in Suzuki et al. In 1988. Here's a paper trail of the various scientists that helped discover this mechanism of Toxoplasma gondii infection. Now we come to this molecule Qa-1b, and this is a very interesting molecule. It is a non-classical major histocompatibility complex, class 1 protein in the class B locus, MHC class 1b. is what we. It is a functional homologue of HLA-E, which is the human leukocyte antigen class 1, histocompatibility antigen alpha chain E. This molecule has limited polymorphism and reduced diversity. And the interesting thing about Qa-1b is that it is expressed on every cell that can express MHC class 1 protein. And what Qa-1b does is it is an antigen-presenting protein and presents leader sequences of MHC class 1 heavy chain. That is, it presents self-antigen. However, the importance of Qa-1b in CD8 plus T cell response is not known. We know that Qa-1b is recognized by CD94 NKG2A inhibitory receptors on NK cells. That is, Qa-1b binds to NKG2A receptors, inhibiting NK cells. And this is thought to have been a negative regulator of CD8 plus T cells in viral and autoimmune infection, as shown in Su et al. In 2017. And this may be important for CD8 plus T cell dependent viral control. And here's a paper trail of showing this molecule Qa-1b and its uses. Notice that how all of the previous experiments have been done in viral infection. Now, the exact function in the immune response of Qa-1b remains very poorly understood. And how immune factors such as non-classical Qa-1b contributing to control of Toxoplasma gondii infection has never been tested before. Therefore, we hypothesized that based on previous viral models, that Qa-1b acts as a negative regulator of CD8 plus T cells in T. gondii infection. Here's a proposed model of our hypothesis. If Qa-1b is blocked, NK cell becomes more cytotoxic and they increase in function. Thus, they target the CD8 plus T cells and kills off and or limits CD8 T cell function. Now here, CD8 T cells are the most important control of T. gondii infection. And in the absence of CD8 plus T cells, mice and or the host, they die from the Toxoplasma gondii infection. Here are the methods that we used. We used an antibody, anti-Qa-1b blockade on mice to test how Qa-1b or the absence thereof affects T. gondii infection. And we used two sets of mice, non-treated and treated with anti-Qa-1b, infected with the ME49 strain of Toxoplasma gondii for 28 days. So we conducted murine survival assays and the parasite burden, and the parasite burden was counted via counting T. gondii tissue cysts in the brain. So here's the experimental model. We used 6 mice for the control, no treatment plus ME49 infection, and 6 mice for experimental anti-Qa-1b treatment plus ME49 infection. And here are the results. After 10 days, the anti-Qa-1b mice had a sharp decrease in survival rate, less than 50%, which is very, in this case, very significant drop in survival. And we can also see that there's a very significant increase of cysts in the brain in the anti-Qa-1b mice harvested. Whereas in the no-treat, there was only about 2,500 cysts in the brain. In the anti-Qa-1b mice, there were over 10,000 cysts. As we can see, only preliminary data was conducted. This experiment has to be repeated. Blockade of Qa-1b was observed to lead to increased mortality of mice and increased cyst burden, leading to a worse infection. And these results suggest that Qa-1b may be important for the generation of adaptive immunity to T. gondii infection. As for how, that is still unknown. Again, I restate my hypothesis here. If we blockade Qa-1b, then we hypothesize that NK cells increase in function and CD8 plus T cells decrease in function, thus contributing to the increased mortality of mice and the increased cyst burden. Here are some future experiments that are planned. We wanted to look at, does Qa-1b affect CD8 plus T cell expression? in Toxoplasma gondii infection? How does Qa-1b affect CD8 plus T cells mechanistically in T. gondii infection? And how does Qa-1b affect the immune response as a whole in T. gondii infection? Unfortunately, we are unable to do further experiments in Qa-1b due to the ongoing COVID-19 pandemic. But this is a topic that many scientists might be interested in doing in the future. Here are my acknowledgements. This is the Gigley Immunology Lab as shown on the right. Special thanks to my PI, Dr. Jason Gigley, and to my GAs, Stephen Denton and Tagataka Roy. And also, of course, special thanks to the Wyoming Research Scholars Program for providing the scholarship for me to be able to do this research. And of course, if you have any questions, please feel free to email me or send me a phone number or something like that. Thanks.